Hello, I'm Tom Martin from the University of California, San Francisco, Helen Diller Family Comprehensive Cancer Center. And I'm joined today with Blood Cancers Today Editor-in-Chief, Dr. Sagar Lonial of the Winship Cancer Institute in Emory, Dr. Shambavi Richard of the Center of Excellence for Multiple Myeloma in the Icahn School of Medicine at Mount Sinai in New York, and Dr. Peter Voorhees of the Levine Cancer Institute in North Carolina. And this is our first roundtable of discussion on BCMA-targeted CAR-T cell therapy. And so we have a second product now. We have a second product of Ciltacabtagene Autoleucel. And Ciltacabtagene is actually a product that originally came from China. And very interesting, we had a patient at UCSF that actually went to China, and he was the first US patient to receive this CAR-T cell therapy. And I tell him all the time, you are responsible for this to be a drug in the US, because he went there with very refractory disease, triple class refractory. We had nothing more really to give him, and he came back without all the extramedullary disease that he went there with, in complete remission, and we were just amazed. We were nervous when he went to China, and we were amazed when he came back. And so, Dr. Richards, can you give us a little update of where we are with the Ciltacabtagene and the clinical development?
Yes, Tom. So yeah, as you said, so the Legend project product and the CARTITUDE studies, I think, reset the bar where BCMA CAR-T cells and just what to expect from patients who were so refractory and so advanced. So CARTITUDE-1 was a phase 1b/2 study to assess the safety and efficacy of Ciltacabtagene. So this is also a second generation autologous BCMA-directed CAR-T product. But one difference, Ciltacabtagene is different from ide-cel in that the tumor binding domain actually targets two BCMA epitopes, and it was designed this way to confer an increased avidity. So patients eligible for this study, very advanced group, again, similar to the KarMMa trials, had to have 3 or more prior lines of therapy, including PI, IMiDs, and anti-CD38 antibodies, or had to be double refractory to PI and IMiD, or had to have progressive disease within 12 months of their last line of therapy. So 97 patients were enrolled and studied on this trial with a median of 6 prior lines of therapy. Very advanced group, 88% were triple class refractory, 42% were penta refractory, and 99% were refractory to their last line of treatment. So the responses happened quick by the first month, and best responses was seen at less than 3 months. And these responses tended to deepen over time. So these were actually updated at the 2-year mark post last patient N. Actually on your paper, Tom, and JCO, and so the results again were outstanding. The overall response rate was 98% for all patients, very deep responses, stringent CR in about 83% of patients. 61 of these 97 patients were MRD evaluable, and 92% of them were actually negative. But even more impressively, these MRDs were sustained in 68% of patients at 6 months, and in about 55% of patients at 12 months. So also impressively, the median PFS, overall survival, and duration of response has not been reached at this almost 28-month follow-up, but at the 27-month PFS rate is about 55%. But then when we teased out patients with the deeper responses, so PFS for stringent CR was 64%, and then for those who had sustained MRD at 6 months, it was 73%. So these were excellent results. Again, by the same token, when looking at 2-year overall survivals were 70% at 27 months, but then when looking at patients who had sustained the MRD for at least 6 months, this was well over 90%. So compared to standard of care approaches where we expected median overall survival for triple class refractory patients to be less than 12 months or penta refractory patients to be less than 6 months, these were indeed impressive results. And based on this, Ciltacabtagene was approved by the FDA last year in February as CARVYKTI, as a commercially approved CAR-T product. Interestingly, again, there were some other differences when looking at Ciltacabtagene in terms of correlatives for response. For one thing, not surprisingly, depth of response was important for ultimate outcome in terms of progression-free survival. So patients who were in stringent CR or sustained MRD negativity had a longer PFS. Interestingly, also patients in the higher risk groups, like the high-risk cytogenetic abnormalities or patients with baseline plasmacytomas, they did have good response rates, comparable response rates, but their PFS was a little bit less as expected. So patients at high risk continue to be at high risk, even with these very powerful tools in terms of high tumor burden or high-risk cytogenetics or baseline plasmacytomas. But interestingly, no correlation was observed with these in terms of expansion of CAR-T cells, exposure, persistence, or even BCMA expression on myeloma cells. So that's something interesting with this product, seems to be a little different. And other correlatives that we heard about recently was that in terms of baseline pro-inflammatory parameters that seem to also influence the progression-free survival. So patients who came in with baseline high ferritins or CRPs or IL-6 and other things, even high baseline neutrophils, things that you think they somewhat in this pro-inflammatory milieu. So these patients also seem to have a shorter progression-free survival as compared to the overall group.
Thank you, Dr. Richards. Are there any clinical features in a particular patient that you'd say, okay, this is a great patient for CARVYKTI or great patient for Ciltacabtagene?
Oh, when I think of, ultimately, I just want to get the best response for the longest time. So and I think it's quite intuitively, patients who just are in a better disease shape, just, in terms of low tumor burden at the time of not progressing at the time of the CAR-T. All of these inflammatory markers they're not really sizzling. They're not, high ferritins and high, CRPs when they come in. So overall, they're in this quiescent state, the disease is behaving itself. They get through the CAR-T process most smoothly, not too many ruffles, not the CRS is managed, not too much of the other neurotoxicity and things like that. And they tend to just do better overall in terms of disease. But I guess we could say that with any tool of therapy.